So now we're going to go into microscopic anatomy of the cell contraction, initiation of cell contraction, and the physiology of skeletal muscle. So a skeletal muscle cell is also known as a muscle fiber. They are quite long, and their diameter is the typical diameter of a cell, like 10 to 30 microns, probably closer to 30 microns in size, but they have that variation. But these can be quite long cells, and because of that, they are actually multinucleated, and their nucleus is going to be peripheral and not in the center of the cell. Inside the cell, we have what's called the myofibril. So the myofibril is the actual contraction unit inside the cell. There's also something called sarcoplasmic reticulum inside the cell, and so this becomes really important in muscle contraction. It's something like an endoplasmic reticulum. It's very similar, and it goes from out here at the nuclei. It will actually travel to the inside and will actually be next to and surround these microfibrils. And there's a system of transverse tubules, also known as T-tubules, that actually come from what's known as the sarcolemma or the cell membrane and actually travels down into the cells. Very important in the physiology of muscle contraction. So a sarcomere is a series of protein filaments that make up a contracting unit. And so when we look at a sarcomere, we're going to have what's known as an A band and an I band. A sarcomere usually goes from basically a line in here called a Z plate from one I band to the next I band, and you can actually cut this I band in half. So one small contracting unit in here ends up being a sarcomere. We put a whole bunch of these contracting sarcomere units together and run it the full length of the muscle, and we have what's known as one myofibril. And there are potentially up to hundreds of myofibrils within a muscle fiber cell. So looking at the sarcomere, it's actually going to be made up of myosin and actin proteins, and this is that part that's actually going to be important for contraction is the sarcomere. It's going to run from one Z disc you see here to the next Z disc, or also known as a Z line. When we actually go down to the molecular level, you're going to have the Z line here, which is actually a protein that's going to hold on actin fibers. And so there's a Z-line protein here, a Z-line protein here. We have actin fibers that come off, and so actin fibers are going to actually be anchored to this. And then free floating between these actin fibers end up being myosin heads. So these are actually myosin proteins, and they actually have these little attachments that come off that look like heads. And what they do is they actually ratchet down the actin fibers, and they actually shorten the length of the cell by actually pulling these actin fibers together, and they will actually almost touch the Z line. Where we have myosin at, the muscle stains darker, and where it's the Z line and just actin, it's going to stain lighter. Hence why we get the striated appearance within skeletal and cardiac muscle. It's really important is to know in skeletal and cardiac muscle, all of these actin and myosin fibers in myofibrils here, they all line up perfectly parallel up and down the cell, whereas in smooth muscle these would actually crisscross, and that's why it doesn't appear striated. Looking at the visible striations, we'll start off with the Z line, also known as a Z disc. So this is a protein fiber that's actually going to be where the actin fibers are going to attach. It's going to be in the clear, what we know as the I band area, and it's going to be right in the center. And a sarcomere runs from one Z line or Z disc to the next, and this will actually be the contracting and shortening units right here. And when you get muscle contraction, basically what happens is all these Z discs actually get pulled in and almost touch all of these darker A bands right here where we have the myosin at. The I band is just where there's just, during relaxation of the muscle, where there's just the Z line and the actin. And these are the lighter staining bands when we end up seeing striated muscle. A band is where we have myosin at. And when we look at the A band, we actually have the A band, and inside the A band, we have the H band. And so the A band and the H band, there's myosin fibers. And the difference between the two is when we get to the H band, it is a light area in the middle of the A band, and it's actually where there's an absence of actin fibers when the muscle is not contracted when it's relaxed. And this is the area where actually actin fibers will almost touch each other when you have contraction. When we have a muscle contraction, the Z band gets pulled this way and gets pulled this way, and the actin fibers almost meet where this H band is right here in the middle. If we were actually cross-section skeletal muscle, when it's actually contracted, it's no longer striated because all of these areas are pulled in and it becomes more homogeneous in appearance. But when it's in relaxation, when we see it, it's heterogeneous. So we have areas where we can see where it's just actin, an area where we have actin and myosin, and that's going to be darker because there's more density here. And then it'll be a little bit lighter again where we just have myosin and no actin. Also looking in here, this is three-dimensional. So we look here and it's very easy to see it two-dimensional, but what you have to see is here is all your different myosin heads and myosin proteins, and here's all of your actin fibers that are going to move on. And then when you put them together, you can actually see how they all work, interdigitate to actually make more of a 3D structure right here. So what causes a muscle contraction? Well, it ends up being a nerve input. And so we have what's known as the neuromuscular junction. This is where we're going to have a teledendrite down here on the synaptic area of the nerve that's going to be right in close proximity, does not actually touch, but right next to a skeletal muscle. And what ends up happening is this synaptic vesicle right here into the neuron is going to fuse with the cell membrane of the nerve and is actually going to then release out the chemical known as acetylcholine. And so acetylcholine comes here and actually binds the receptors on the side of the skeletal muscle, and that causes the skeletal muscle to go through a chemical change and allows for the muscle to contract. We have to stop that contraction, and how that actually happens is the chemical acetylcholinesterase actually breaks down acetylcholine and removes it from the receptor. We have motor units, and so one nerve fiber and all the muscle fibers it innervates is one motor unit. So when we look at a nerve, it's going to sit there and actually have an input into multiple muscle fibers. We can operate these motor units individually to do small, delicate movements like play the piano, or to sit there and manipulate something really small like trying to put a tiny screw into your glasses. We can have multiple motor units work together, and when we put multiple motor units together, they end up moving large groups of muscle fibers. And so this is where we can get large, powerful movements like sitting there and moving your knee while you're running, or flexing your elbow to lift up a pencil. So looking at connective tissue layers again, when we look at motor units, we can actually work down here at the fascicle unit inside the muscle, and this is where a lot of times the nerve will actually go to innervate. And so a fascicle is going to be multiple muscle cells and fibers actually held in together. It's going to be surrounded by what's known as endomysium, so that actually holds the fascicles together. When we go into a larger group to actually make the full muscle itself, we're actually going to connect multiple fascicles together and multiple endomysiums. And so we have connective tissue within the inside of the muscle called perimysium, and this actually holds all the epimysiums and fascicles together. And then lastly, we have the epimysium on the outside, and so the epimysium is the connective tissue that actually holds all of these muscle cells together. Endomysium, perimysium, and epimysium will actually be continuous into turning into either a tendon where it attaches to the bone, or an aponeuroses where we end up having an attachment like at the linea alba. Muscle is very highly vascular, so there's a lot of blood vessels that come in here, and it's very rich with nerves because nerves have to sit there and actually hook up to tell these muscles what to do. So actually looking now at contraction and relaxation, so when we're looking at that whole muscle right there, how it actually works is you're going to have the nerve impulses come down to the individual motor fiber units, so the individual fascicles. They're going to reach the end of that nerve fiber, and the acetylcholine is going to be released into the synaptic space there, and it's actually going to bind to the surface of the muscle cell known as the sarcolemma, same thing as your plasma or cell membrane. We just happen to call it a sarcolemma in muscle. The impulse from this acetylcholine is going to travel across the sarcolemma and go down through what's known as the T-tubules, and it's going to go to the interior of the cell. When the T-tubules are stimulated and they travel to the inside of the cell, they're actually going to reach the sarcoplasmic reticulum with their signal from the acetylcholine, and this in turn causes the sarcoplasmic reticulum to release calcium ion into the sarcoplasm. So it's the same thing as the cytoplasm. So calcium is going to be released. It's stored in the sarcoplasmic reticulum. It is now going to be released into the sarcoplasm where the actin and myosin fibers are at. And so calcium actually goes to those myofibrils, and it actually is what initiates and starts the contraction of the myosin, and the myosin ratcheting and walking across the surface of the actin. Some energy is required for this, and that's going to be supplied through ATP. After contraction is finished, the cell has to put the calcium back in to the sarcoplasmic reticulum, so it's going to be actively pumped out of the sarcoplasm and return to the sarcoplasmic reticulum. And so this is very, very energy intensive, and so this requires requires a lot of ATP. So when this occurs and muscle stops its contraction and goes back into relaxation, the muscle is going to return to its original length. So when we look here at a muscle fiber in relaxation, and we're actually looking at the sarcomere, here we have the myosin and here we have the actin fibers. And so when you have contractions, once again, these myosin fibers, they have the heads, there's little spots here on the actin fibers, are actually going to sit there and ratchet their way down, and they're going to pull the Z lines physically close together. And this is also known as a cross-bridging action of the ratchets right here. And that physically shortens the actual sarcomere. And this goes across all the way up and down the muscle fiber, which actually shortens the length of the entire muscle. Key to know is the all or nothing principle. So when stimulated, the muscle cell is going to contract all of the myofibrils and all of the sarcomeres, and they all contract completely, or they do not contract at all. They're either going to accept the stimulation of acetylcholine from the nervous system and initiate the contraction, or they're not. They have enough calcium in the sarcoplasmic reticulum to start a contraction all the way down the muscle, or they do not. So it does not go halfway. It's an all or nothing. And there is a cycle for actually doing this contraction, and it's what we call a twitch contraction. And so this happens very fast, and it's actually a very coordinated motion. On some of the myofibrils are going to contract at one time, and the individual muscle cells. So we might have like one fascicle contracting, and then there's going to be an alternation of another fascicle contracting as you're moving or doing a muscle. This happens really, really fast, and so we start off with what's known as the latent phase, and this is a brief hesitation as contraction is started. This lasts 0.01 seconds or 10 milliseconds, so it's very fast, faster than actually blinking your eye. Next we get into the contracting phase, and so this is where the sarcomere is actually going to contract and the full muscle cell or fiber is going to contract, and this is going to last 0.04 seconds or 40 milliseconds. Lastly, we get to the relaxation phase. So this is where the calcium is going to be put back in the sarcoplasmic reticulum to get ready for the next contraction. So the sarcomere actually is going to relax and the full muscle cell will relax and gets ready to repolarize for the next contraction. And this lasts 0.05 seconds or 50 milliseconds. In total, this full twitch contraction right here to take place is a tenth or 0.1 seconds to go through this full cycle. So if you're doing something like trying to pick up a heavy weight, your muscles do this multiple times per second. They will actually do this 10 times per second, go through this twitch contraction series right here. So when we look at contraction efficiency, this is going to be the nerve pulses actually all come a tenth of a second apart, 0.1 seconds. And so you get these complete muscle fiber twitches that come from it. And it's this smooth, sustained muscle contraction that comes out of averaging out the activity of having some muscle cells contract while others relax. So they don't all contract at the same time. That's what they mean out of sync. They're slightly asynchronous from each other to make it to where it's smooth. Sometimes we have issues with this. So diseases like Parkinson's disease, do excuse me for not correcting this on the slide. I just caught it. I had a slightly dyslexic moment, but that should not be APT, but should be ATP, adenine triphosphate, is the energy for all activities in cells. And so while calcium actually causes the sliding of the actin and myosin fibers there, ATP is important for actually putting that calcium back up into the sarcoplasmic reticulum. And actually the initiation of releasing the calcium out of the sarcoplasmic reticulum also comes down from ATP. ATP will actually go to ADP, adenine diphosphate. And so there is a chemical within muscle known as creatinine phosphate that helps to rapidly convert ADP back to ATP. There's a lot of energy that's actually used in muscles, so there's going to be a lot of glucose and oxygen used to help convert this ADP back to ATP and to help out with this creatinine phosphate doing its job in that conversion. So muscles actually store extra energy of glucose in the form of glycogen, the same way that extra glucose in the body is stored in the liver as glycogen. Extra oxygen is actually stored in muscle in the form of myoglobin, which is very similar to hemoglobin and actually gives muscle its red appearance. When we look at doing muscle contractions, we can have aerobic and anaerobic metabolism. So aerobic metabolism has adequate oxygen to meet the needs of the cell, and it's where we have our maximum energy and maximum efficiency of muscle cells. Muscle cells that continuously go through contraction and are doing heavy work, though, can run out of oxygen. There's not enough oxygen in the bloodstream to keep muscle a lot of times fully fueled, and there's sometimes not even enough glucose in the blood supply. And so when the cell starts to run out of its oxygen from its myoglobin, it can actually go into anaerobic metabolism, and so it starts making lactic acid, which is due to an incomplete breakdown of glucose and a lack of the Krebs cycle due to a lack of oxygen. Big thing with anaerobic metabolism and lactic acid buildup in muscles is this is actually what makes muscles become fatigued, sore, and painful. And too long of anaerobic metabolism in the muscle can actually cause death of the individual muscle fibers. Key to all this ATP generation and all this energy use is heat. So heat is going to be a byproduct of muscle activities. The body can actually use skeletal muscle in the form of shivering to actually try to raise body heat, but the body also can generate too much body heat through muscle contraction. And in those cases, the body has to have ways of getting rid of that excess heat. So this is where we get like panting in dogs or sweating in people and horses. That was an overview of the microscopic anatomy and then of the muscle cell and then the physiology of contraction. Next, we're going to look at cardiac muscle.